Hello, my name is Derik Haggard, and I am a postdoctoral research fellow in the Center for Computational Toxicology and Exposure at the U.S. Environmental Protection Agency. The title of my poster is Optimizing Transcriptomics for High-Throughput Bioactivity Screening of Environmental Chemicals. Before I begin, I need to state that the views expressed in this presentation and the poster are my own and do not necessarily represent the views or policies of the agency. The primary motivation for this work revolves around the push to develop new approach methodologies, or NAMs, for chemical safety and risk evaluation, with the goal to replace existing low-throughput in vivo methods with high-throughput and high-content in vitro assays. High-throughput transcriptomics, or HTTr, shows promise as a first-tier screening approach due to its ability to detect numerous mechanisms of bioactivity, and the U.S. EPA has begun development of an HTTr screening assay using a targeted RNA-seq platform. In this brief presentation, I will describe the screening assay and present findings from a large chemical screen in MCF7 breast cancer cells, with a primary focus on the reproducibility and stability of the reference standards that we use in this assay. Dose plates are prepared with a set of test chemicals. Test plates are first seeded with cells, then chemicals are transferred from the dose plates to the test plates in random arrangements using the LabSite Eco liquid handler. Additional QC standards are then added to each plate, which include reference RNA, lysis buffer blanks, and bulk lysate preparations taken from cells exposed to DMSO vehicle control or trichostatin A. In addition, parallel test plates are prepared in the same manner, and an apoptosis cell viability assay is performed to determine cytotoxic chemicals or cytotoxic concentrations for chemicals. All subsets of test chemicals are plated in triplicate, one replicate per test plate, using independent cell culture batches. One cell culture batch can seed a total of about 10 test plates. A plate group is defined as the set of 3 test plates that a given chemical has been added to. In total, the HTTr MCF7 screen consists of 48 plate groups that were screened across 4 independent experimental blocks, separated by roughly 1 month between blocks. Sequence alignment of FASTQ files were performed using HiSat2, and uniquely aligned reads were quantified using SAM tools. Multiple quality control metrics were calculated for all samples, and samples with sufficient quality were further analyzed using DE-Seq2 and gene set enrichment analysis using single sample GSEA, or SSGSEA. A total of about 2,200 chemicals were screened as part of the HTTr MCF7 screen, representing a little over 54,000 individual samples. Here, we demonstrate the overall quality of all these test samples, which are separated by sample type for 3 quality control metrics. Panel A represents the total mapped reads of all samples, with a minimum depth of 300,000 reads as our threshold for sample quality. Panel B represents the mapped fraction of reads across samples, with a minimum mapping rate of 50% as a threshold for sample quality. And panel C represents another quality metric, which we call NCO5, which is the total number of probes that have greater than or equal to 5 total counts for each sample. Overall, we found that using these quality metrics and others that are not described here, a total of 98% of all samples were considered as having sufficient quality. In addition to the quality control metrics shown previously, we also performed correlation analysis using log counts per million and with DE-Seq2 moderated fold changes for all reference samples. Here I will describe the results for the reference chemical treatments, trichostatin A, genistein, and sirolimus. Please refer to Figure 3 for a summary of the results for the reference RNA and bulk lysate samples. The figure on the left shows the distribution of the average correlation of log counts per million for the 3 reference chemical treatments and the DMSO control. Also known as the D statistic, this measure can be used to identify additional potential outliers that pass the initial quality control metrics that we calculate. We use a threshold of 3 standard deviations from the mean D statistic, denoted by the vertical dashed lines in the figure, to flag outlier samples. Overall, we observed high correlation across the log counts per million for all reference samples. The figure on the right is a k-means clustered heat map of DE-Seq2 log fold changes of the 3 reference chemicals. We observed strong separation of samples based on reference chemical treatment, suggesting good reproducibility of gene expression profiles across all plate groups. Finally, we performed SSGSEA to determine whether the gene expression profiles of the reference chemicals were enriched in their known molecular target. Genistein is an estrogen receptor agonist, sirolimus is an mTOR PI3 kinase inhibitor, and trichostatin A is a histone deacetylase inhibitor. The figure on the left demonstrates the gene set level approach in blue results in much higher correlations compared to a probe level fold change correlation approach, which is shown in pink. The figure on the right shows the distribution of SSGSEA signature scores for gene sets, or what we call signatures, specific to the molecular target associated with each reference chemical. As shown, the reference chemicals had much larger signature scores for their known molecular targets compared to each other and a random set of gene signatures. In summary, we have demonstrated the stability and reproducibility of the reference samples within the HTTr assay. Roughly 98% of all samples passed initial quality control, suggesting good performance of the experimental method. An additional analysis showed good correlation at both the counts per million and the DE-seq2 fold change level for all reference samples. Gene set enrichment analysis also demonstrates aggregate level methods result in higher treatment correlations compared to probe level methods, and that the expression profiles of the reference chemical treatments were enriched in their specific no molecular target, suggesting that the HTTr assay can identify specific mechanisms of bioactivity. And with that, I would like to acknowledge all of the many colleagues and collaborators that were involved in this work, and also thank you for your attention.